When we talk about genetics, there are a variety of inheritance patterns. And when we talk about dominance, dominance does not always work exactly the way that we've talked about so far. So the relationship between genotype and phenotype is rarely as simple as what we've been describing it. Now, there are different types of dominance, so we call this a spectrum of dominance. And what we've talked about so far in Chapter 14 would fall under the category of complete dominance. Complete dominance means that the phenotype of the heterozygous individual and then the dominant homozygous individual, they're going to be identical to each other, so that you visually could not distinguish them from each other. So if we look at this example with flower color, the capital P represents the color purple and lowercase p represents white. If you have two capital P's, they look purple. If you have one capital P and one lowercase p, they look purple. And notice again that they are exactly the same shade of purple. So this is really why we had that test cross back in the beginning of Chapter 14. The test cross was a way that we could take something that looked dominant, something that looked purple in this case, and we could go a step further, do a cross, and actually figure out what the genotype of that particular individual was, whether they were homozygous, whether they were heterozygous. So with complete dominance, the dominant allele completely covers up the phenotype of the recessive allele. That is not always the way that dominance works. Another type of dominance is what we call codominance. When we have codominance, there are two dominant alleles, and they affect the phenotype in separate, distinguishable ways. So this means that if you are heterozygous for those two different dominant alleles, you would see the effects of both of them. A good example of codominance is blood types. So when we talk about blood types, there is an allele for type A blood, and there is an allele for type B blood. And if you have the A allele and the B allele, you have type AB blood. So notice that both of them are going to exert their influence on the phenotype. Now we will come back to blood types and we'll work some crosses with regards to blood types a little bit later in the chapter. The last type of dominance that we want to introduce is what's called incomplete dominance. Now, incomplete dominance is a little bit like blending, because in this case, the phenotype of the F1 hybrids is going to be intermediate between the two true breeding homozygous parents. If we look at this example here from snapdragons, we've got two different alleles. We have a CR allele and we have a CW allele. The CR allele is for red and the CW allele is for white. So if you have two copies of CR, so homozygous CR, that produces a red flower. If we have two copies of CW, that produces a white flower. Now, if we were to take these parents and cross them with each other, from the CR parent, we're always going to get a gamete that has CR, and from the white parent, we're always going to get a gamete that has CW. So if we put these together to get the F1 is going to be heterozygous, but as you can see in this picture, it doesn't look like either one of the two parents. We had a red parent and a white parent, and now we have an offspring that is pink. So this is what we mean over here when we say that the phenotype of the F1 hybrids is somewhere in between what the phenotype of the two parents was. So notice that there would not ever really be a need for a test cross in this case when we're talking about incomplete dominance, because if we have a heterozygous individual, visually we're going to be able to distinguish that because they have their own unique look to them. Now, we don't really consider this blending as we described at the very beginning of Chapter 14, because in that case, the alleles were actually blending together and becoming a new version of the gene, for example. So this would be like the CR and the CW mixing together to become, say, a CP for pink. Not the case. So when that pink flowered plant is crossed with another pink flowered plant to produce the F2, it's going to really work the same way as before. Half the time we're going to pass on the CR, half the time we're going to pass on the CW. So if we draw the Punnett square here and we fill in the boxes, notice that we can get some red flowered plants back, we can get some white ones, and then we do have some pink ones there as well. Now in this case, the phenotypic ratio of the F2 generation is going to be a little bit different from what we had when we were talking about complete dominance. When we're coming up with the phenotypic ratio, we want to list the phenotypes. So we have red, we have pink, and we have white. And then we count up how many we have of each one. So this is going to give us, in this case, a 1 to 2 to 1 phenotypic ratio. Keep in mind that when we talked about the phenotypic ratio of the F2 with complete dominance, we had a 3 to 1 ratio. So there is a difference there. If you knew that you were dealing with a single gene and 2 alleles and you had 3 phenotypes possible, that should clue you into the fact that maybe you are perhaps dealing with a gene that has incomplete dominance. Now it's very gene specific. So for flower color in snapdragons, incomplete dominance is what does apply. For flower color in Mendel's pea plants, they always had complete dominance. So it is specific to a specific gene. We look at a question here and try to work out a cross with this example. If you were to cross a white snapdragon with a pink snapdragon, we want to know what fraction of the offspring or progeny will be white. Okay, so we want to first write our parents' genotypes. We've got a white snapdragon, which would be this genotype, and we're going to cross that white one with a pink one, which would be this genotype, and we want to figure out the fraction that's going to be white. Okay, so if we draw our Punnett square, we're going to put one parent's gametes down the side. I'll put the white ones over here. And we're going to put the other parent's gametes across the top. So if we fill in the boxes, we look at this, we can see that this one would be white. This one right here would be white as well. So we would expect that 2 out of 4, or in other words, 1/2 of the offspring from this cross would be white. We would also have 1/2 pink offspring, which would be these over here. Okay, so make sure we understand with incomplete dominance, we're not working the Punnett squares out any differently than what we did before, but the heterozygous ones now have their own unique look. Now, if we talk about why all this works, alleles, we must keep in mind, are variations in DNA sequence. So these are just sequences of DNA, and their purpose is really to tell the cell how to make something. They are going to provide the instructions for the cell to make a piece of RNA through the process of transcription, and that piece of RNA is going to tell the ribosome how to make a protein through the process of translation. So when we're talking about alleles, they don't physically interact with each other. They stay on separate chromosomes, homologous chromosomes, but the product, the actual protein that is produced from that sequence, is going to be a little bit different. And because it is a little bit different, that's going to contribute to a different phenotype inside the cell. So when we talk about our flower color example, this CR allele tells the cell how to make a red pigment. Well, the CW allele is basically no pigment. So if you think about the fact that in CR, we have 2 copies of instructions to make red pigment, so you get a lot of red pigment that's actually made, and that contributes to the red flower color. If you have 2 copies of no pigment, you have no pigment and it appears white. When we're talking about the heterozygous one, here we're making red and here we're making nothing. So really what we're getting is not a different color, but we're getting half as much pigment as what we had up here. And because we have half as much pigment, we see a diluted red, which then appears pink to us. So we do just want to keep in mind that the alleles tell the cell how to make something very specific, and whatever that product is, the protein, that is going to determine the overall appearance or look of the individual. Now, if we continue on talking about dominant and recessive, we do want to emphasize that dominant alleles are not necessarily the most common alleles in a population. That is a very common misconception that students have. So, for an example we've already talked about, widow's peak, the little point in the hairline, that is caused by a dominant allele. However, having a widow's peak is not the most common phenotype that you see. So most people do not have a widow's peak, which would mean that most people do have the recessive phenotype. If we look at a few other examples, dimples, this can be chin dimples or cheek dimples. That is caused by a dominant allele. And while that's not necessarily rare, the majority of people do not have dimples. So that means that the majority of people do have the recessive phenotype to them. Another less common example is what we call polydactyly. Polydactyly is when there is an extra digit, and this extra digit can be on the hand or the foot, and this is caused by a dominant allele. Now, when we have something like polydactyly that is fairly rare, when someone has it, we kind of make the assumption that they're going to be heterozygous. This is because if they were to be big A, that means that both of their parents would have had to contribute a big A to them. And that's not real likely that that's going to happen. So if someone has polydactyly, we assume that they are heterozygous. If they're heterozygous, keep in mind that half the time they're going to give their child a big A, and half the time they would pass on a lowercase a to their child. So if half the time they're passing on this allele, we would expect that 50% of their children will also have polydactyly. Now, we don't see too many people that have this condition anymore because very shortly after birth, they tend to remove the extra digit. Keep in mind that just because we can alter the phenotype, we remove the extra digit, we have changed the appearance, we are not able to change the DNA. So just because it outwardly does not appear that there is an extra digit there, genetically, that has just as high of a probability as it ever did of being passed on to the offspring. Now, while you may or may not have seen this in people, this is also the same character that gives cats a sixth toe. So if you've heard of the Hemingway cats, or you have one, or you've seen one, many of those have a sixth toe on them, and that is from basically polydactyly in cats. We also want to talk about the fact that, generally speaking, for most genes, there is not just a capital A and a lowercase a. Most genes exist in populations in more than 2 allelic forms. So that means that there may be 10 different A's that are possible. So we might have an A sub 1, an A sub 2, an A sub 3, and so on. Now, just because we may have a lot of different A alleles possible, that does not change the number of alleles that we will inherit or that we will have in our DNA. Keep in mind that as humans, we are diploid. And diploid means that we have 2 chromosomes. So those 2 chromosomes will contain 1 allele on each one of them, which means that for the A gene, we are going to get 2 alleles. Those may be 2 of the same allele. They may be 2 different alleles. Now, a good example of a gene that has multiple alleles possible is when we talk about blood types. And in this case, we're just talking about the letter for the blood types, so A, B, and O. This gene has 3 different alleles, and what these alleles do is they tell the cell how to make a protein that is going to stick to the outside of the red blood cells. If we have the IA allele, this one produces a protein that has a characteristic shape, and we're going to call it a triangle shape. The IB allele sequence is slightly different, and it makes a slightly different protein that now has a circular shape. And the little I allele does not produce the protein. So there are 3 alleles possible in the human population, but every individual is going to have 2 because they are diploid. So if we look at all the combinations that we can get with these 3 alleles, we do have 6 different genotypes that are possible. And if we look at them here, if you have 2 copies of IA, then you're going to be producing a lot of the triangle protein. So we would have red blood cells that have triangles on the outside. That's what we call type A blood. If you are heterozygous in this condition, so if you have the IA and a little I, because the little I doesn't produce protein, the red blood cells are really going to look exactly the same. So that would also be someone that has type A. With type B blood, the genotype homozygous for IB is going to produce lots of circles. That's called type B blood. And then if you're heterozygous and have IB and a little I, then you're also going to have red blood cells that have the circles on the outside. So that would also be type B. If we have IA, IB, then we're going to get circles and triangles on the outside of our red blood cells, and that's what we call AB blood. This is where we have the codominance example. So notice that the IA is not covering up the IB and vice versa. We see the effects of both of them being there. If we have two of the little i's, that's going to be what we call type O blood. So there's none of these particular proteins on the outside of it. Now, if we talk about blood transfusions and who can accept blood from who, basically our bodies attack things that look foreign to them. So if you were someone that had type A blood, your body is used to seeing type A blood, which means it's used to seeing triangles. So if you had type A, you could certainly receive blood from type A individuals. The type B blood and type AB blood would look odd because they have the circles, which is not something that's naturally produced in that person. So those would not be acceptable things to give to someone that was type A. But type O blood would work because it doesn't have the proteins on the outside. So someone with type A blood could accept blood from either A's or O's. Someone that's type B blood could accept from B's or O's. The AB person could accept any of them. So AB is what we typically call the universal acceptor. And then the type O, they can only accept from type O. However, type O can donate to any of the other blood groups. Now, if we start talking about positive and negative blood types, that is another gene that's involved. So that's another layer of complexity that's on top of this. So we're not going to really throw that into the mix here. We do want to be able to work out Punnett crosses, Punnett squares involving blood types. And just keep in mind that if you do have, say, a person that has type A blood, we don't necessarily know which of these two genotypes they might be. So you may have to have a little extra information there to sort it out, or if it can't be sorted out, then you would have to consider both of the possibilities. Same thing for someone that has type B blood. We talk about some other cases with regards to genetics. Pleiotropy is when we have one gene that has multiple phenotypic effects. A good example of this is sickle cell anemia, which is something we've looked at a number of times already. Sickle cell anemia is caused by a mutation in a single gene, so a change in the DNA sequence, which does affect the shape of the protein. Now, the shape of the protein itself is a phenotype. The fact that it changes the shape of the red blood cell is a phenotype. But then really, if someone has sickle cell anemia, it is not so much that the blood cell looks different, but we have a lot of other things that result from that. So if you look at all of these symptoms of someone with sickle cell anemia, they can have breakdown of red blood cells, they can have anemia, heart failure, paralysis, kidney failure. So notice there's a lot of things listed there, and all of those things would be considered phenotypes of this particular gene. So when we're talking about pleiotropy, it's one gene that causes, say, a whole list of phenotypes. This is very common when we're talking about genes that result in genetic disorders or diseases. So sickle cell anemia is just one example. You do not have to be able to work out crosses involving pleiotropy, but we do want to recognize if we're presented with an example of pleiotropy. So if you have a single gene that is causing a whole bunch of different phenotypes, that would be pleiotropy. Another special case is what we call epistasis. Epistasis is when you have a gene at one locus that alters the phenotypic expression of a gene at a second locus. So to look at a couple examples here, we do have a mouse color gene, and we have the color is the B gene. So we can have capital B and lowercase b. Capital B codes for black, and lowercase b codes for brown. Okay, so very simple. Black is dominant, brown would be recessive. However, we've got this second gene, which is the C gene, and we've got capital C and lowercase c. And what the C gene codes for is pigment deposition. So basically, does the pigment get placed in the hair or not? With capital C, the pigment is deposited. And when we talk about lowercase c, it's not deposited. Okay, so if we look at how all of this works, we've got two black mice up here that we're going to be crossing with each other. Notice that both of them have a capital B, which codes for black, and they both have a capital C, which means that the color is going to get deposited in the hair. So they look black. If we look at all of the boxes in this Punnett square, notice that for any of the black mice, they have at least a capital B, one capital B, and they have at least one capital C, which says, yes, the pigment gets deposited in the hair, and now we see that they are black. If we have brown mice, they have two lowercase b's and at least one capital C, so they have brown, and it gets deposited. And then if you look in these bottom four boxes, or this corner here, we've got some white mice. These mice are white because they have this combination of two lowercase c's. If we have two lowercase c's, that means that the color, whatever it happens to be, is not getting deposited in the hair. And if it does not get deposited in the hair, it really doesn't matter what the B gene is or what the alleles we have for the B gene are. So when we're talking about epistasis, in this case, you've got this C gene that controls whether we even get to see the phenotype of the B gene. If we look at a more common example, Labradors, they have the exact same system working. In this case, we have black and brown, so capital B and lowercase b, and now instead of a C gene, we call it an E gene. Capital E is for deposition or depositing of the pigment, and lowercase e is for not depositing the pigment. So if we have black Labradors, we have at least one capital B and one capital E. If we have chocolates, we have two lowercase b's and at least one capital E. And then the yellows, those are where we actually have the pigment not getting deposited into the hair. We also, just like with pleiotropy, we want to be able to recognize examples of epistasis so that if we know that we've got two genes and one of them is controlling whether or not we see the phenotype of the second one, we're going to be able to pull out that epistasis term. Now, if you look around you, many human appearances, there's not just two possibilities. So these vary in the population along what we call a continuum, and we call these quantitative characters. So for example, if we were talking about height, we can't just score people as being either 4 foot or 5 foot. They're not 4 foot or 6 foot. They can be any possible height. So that's what we mean along a continuum. The reason they vary along a continuum is because we have polygenic inheritance. Polygenic inheritance is when you have an additive effect of multiple genes on a single phenotype. So there are many genes involved in controlling height or quantitative characters. What we're talking about here is really the exact opposite of pleiotropy. So we do want to make sure we can distinguish the two. With pleiotropy, we had one gene and many phenotypes. Here we have many genes, one phenotype. If we were to survey a bunch of individuals for a quantitative character, in this graph we're seeing a survey of skin color. So skin color is another good example of a quantitative character or something that varies along a continuum. We don't really have a white and a black. We have all different shades of tan and brown. So if we were to do a survey on a population of individuals for skin color, we would find that we have a few individuals that are super light, we have a few individuals that are super dark, and most individuals are going to be somewhere in the middle here. So they're going to be a medium color. For quantitative characters, we get a bell-shaped curve, as you see in this diagram, when we survey phenotypes. Height would be another good example. So we've got some people that are extremely short, some people that are extremely tall. Most people are more of a medium height. Quantitative characters vary along a continuum because they're controlled by many genes. Now, many phenotypes are also affected by the environment. So it's not strictly a genetic component that determines phenotypes. So in this example, the pictures that you see here, these are hydrangea flowers. And hydrangea flowers, at least the older varieties of hydrangea flowers, they are very definitely influenced by the soil that you plant them in. If you plant a plant in acidic soil, it produces a certain color of flower. If you dig it up and you move it to a basic soil, it produces a different colored flower. So you've got the same plant that's changing the color of the flowers that it produces depending on its environment. So phenotypes can be affected by the environment. They can change. Skin color is a good example of that as well, because hopefully we're all aware of the fact that we have kind of a certain skin color. That skin color can be affected by how much time we spend in the sun versus how much time we spend indoors. During the summer, we may have a darker skin color than what we have in the winter. However, we all possess what's called a norm of reaction. And what that norm of reaction is it is a phenotypic range of a particular genotype that is influenced by the environment. So if we go back to our skin color example, everyone has a certain window of skin color for them. So if they stay inside all of the time, they will only be so light. And if they go outside all of the time, they will only be so dark. So I will never get as light as some people. I will never get as dark as other people because I have my own little norm of reaction or phenotypic window that I will be somewhere within depending on the environment that I subject myself to. And that norm of reaction is determined by my genotype. Height is another good one. You may have very tall parents, but if you are malnourished, you're probably not going to get to the top of your height potential. Okay, but if you have very good nutrition, then you probably will get to the top of that height potential. So we have what are called multifactorial characters, and these are things that are influenced by many factors. They have both genetic components, they also have environmental components, and those interact together to produce the phenotype. If we go back and we kind of redefine phenotype, initially we said that phenotype was the visible appearance of something. Well, phenotype does include the physical appearance, but it is a little more extensive as far as the definition goes. It's physical appearance, but it's also internal anatomy, physiology, behavior. There's a lot of things that could fall under that phenotype character or category. But what determines phenotype is the overall genotype as well as the unique environmental history. So the environmental history is definitely going to impact the phenotype or what we see. If you think about identical twins have exactly the same genotype, but if you've known people that are identical twins, they're not exactly the same. So there are many things that differ among them. And a big part of that is the fact that they have different environmental histories. So that does affect the phenotypes. Now, as far as genotype is concerned, there is no difference in genotype from when we initially introduced it. The genotype is the organism's entire genetic makeup. We receive that genotype when we are first conceived. So when the egg and the sperm come together, that brings the alleles that make us up. So that is not something that can really be altered throughout our lifetime.